Certain psychiatric disorders, for example, depression and anxiety, are found amongst different species and can have a great effect on the emotional stability in humans and even organisms like model rats. In determining whether these disorders are caused by mainly environmental factors or neurodevelopmental complications, an experiment was conducted using both low responder rats, also referred to as LR, and high responder rats, which are HR rats, given in which the LR were selectively bred and are already predisposed to psychiatric disorders. We know that there are differences among the two groups' limbic brain, affecting the genes associated with metabolic activity using transcriptome profiling. We hypothesize that this particular region of the brain is important in brain circuit development and synaptogenesis. We also know that HR rats there is a higher COX activity, COX being the enzyme cytochrome C oxidase, which is very important for ATP production and metabolic functioning. Each control group of rat pups had specific genes in the hippocampal region of the brain altered at different time periods after birth to then determine if the timing of the gene alteration after birth proved to have an effect on the ATP production, brain circuit development, and emotional stability of the rats. After altering the genes, we concluded that the rats had an alteration of genes 2 weeks after birth or less, had a predominant disruption in limbic brain development. This disruption in development is likely the cause of lower COX activity and lower metabolic activity in the LR rats and may be linked to more severe psychiatric disorders in later life.
Depression and anxiety are disorders that are seen in humans which can be largely affected by predisposed variety in temperament and emotional reactivity among the population. While it is difficult to perform human experimentation, our lab uses a rat model to easily look at these disorders and behaviors. Our rats were bred to show low behavior response, which can be connected to high depression rates, and high behavior response, which is linked to less depressive-like behaviors and higher energy levels. In this experiment, many genes were altered in the hippocampus of these developing high response and low response rats in the first several weeks of their lives, specifically on days 7, 14, and 21. The timing of these alterations had a large impact on neurological function happening in the brain and COX activity. Genes associated with synaptogenesis and cellular metabolism were marked. These are both believed to be associated with anxiety and depression disorders. Disorders such as depression and anxiety show a variety of similar symptoms when compared to metabolic disorders like fatigue, cognitive deficits, and psychomotor retardation that can be seen in human models, so looking at these factors in our rat models is beneficial to the study. ATP is necessary for cellular processes in the brain. ATP synthesis relies on electron transport chain running smoothly. In the electron transport chain, there's a specific enzyme called cytochrome C oxidase, also known as COX, which is a rate-limiting enzyme in the ETC. This enzyme will be of great significance in this experiment because it is proportional to ATP production and is also used when looking at metabolic activity in several regions of the brain. In previous studies that we conducted, we looked at these emotional behaviors in the hippocampus and amygdala. The differences seen were linked to many functional classes of genes. In this study, we were building on our previously found information and looking at the metabolic changes in the limbic brains of these rats and how this relates to anxiety and depression. The hypothesis that we came up with for this experiment is that we would see specific gene expression and metabolic pathways in the rats displaying a high anxiety-like behaviors, and this would be seen through techniques such as transcriptosome profiling and COX histochemistry.
Using 60 male and 60 female rats, the rats were bred with one another to find the highest HR and lowest LR exploration rats, and then bred again with those rats possessing similar traits to obtain LR and HR control groups. At different time points, 1 week, 2 weeks, and 3 weeks after birth, the rats were sacrificed and decapitated to look at brain regions. The brains were preserved and frozen using a cryostat to enable the comparison of normal developing juvenile rat brains, adult rat brains, and our rat brains using the stereotypical atlas. Using transcriptome profiling, we were able to identify the hundreds of differing genes, including the differing COX activity in the hippocampal and the amygdala regions of the brain.
There are many differences seen in the early hippocampus and amygdala of these HR and LR rats. The bottom graphs in Figure 1 show HR data in red and LR data in green. There is a degree of variability in this data, specifically in the phenotype of the different developmental ages. In the amygdala samples, the HR and LR rats clustered in different regions depending on the developmental ages. There is visibly a larger overlap between the HR and LR rats in the samples taken from the hippocampus. In these samples taken from the hippocampus and HR rats, there are clusters at each time point, but in the P14 and P21 data, the clusters are more close together and not as geographically similar to the P7 region. In Figure 2, we see data from an enrichment analysis test. This figure has to do with mitochondria and cellular metabolism and how they are altered in the HR and LR amygdala and hippocampus. In Figure 2a, we can see that in the amygdala, P is less than 0.05 and FC is greater than 1.5, and in the hippocampal region, P is less than 0.05 and FC is greater than 1.5. In Figure 2C and 2D, we can see that overall the amygdala showed more differences in metabolic genes when compared to the hippocampal region. In the amygdala, an average of 25% of genes in a given metabolic pathway were differently expressed between the HR and LR rats in the first few weeks, while in the hippocampus this was true of only 10%. We can see from the data that P14 was the time where the greatest number of changes occurred in the hippocampus of the HR and LR rats. Figure 5 shows color maps of the COX activity in the limbic regions of HRs and LRs along with time points. In these color maps, a black square shows that the data was statistically significant, while a gray square means that the data was not significant. Data shows that COX activity strengthened as development went on regardless of HR and LR phenotype. There is little correlation of the brain regions in the earliest stages. It can be seen that HR and LR groups showed similar average correlation coefficients at P7, and at P14, HR showed higher correlation. At P21, there was little difference. In Figure 6, we see multidimensional scaling analysis of COX activity differences in the first weeks of life. Here, differences between two brain regions show their association strength. The highest level of divergence happened at P7. Here, the CA molecular level and amygdala showed the lowest levels of COX activity in HR compared to LR. At P14, differences can only be seen in a few places. At P21, all of the brain regions clustered, showing similarities in metabolic demands in these HR and LR rats.
After conducting the experiment, we found that there were many hundred differing genes between the LR and HR rat brains using transcriptome profiling. We noticed significant differences among the groups when looking at the early postnatal limbic brains, specifically the amygdala and the hippocampal regions in the brains. Looking at the data showing the significant changes in the gene expression, our next question is, when did the HR and LR brains develop differently? And specifically, what parts of the brains are responsible for these predominant phenotypic changes? In the P7, being the rats that had their genes altered 1 week after birth, and the P14 data, being the rats that had their genes altered 2 weeks after birth, there were hundreds of genes that differed between the two control groups in the amygdala and the hippocampus. For those rats in the P21 data set having their genes altered 3 weeks after birth, there were over 1,000 genes altered between the two in comparing the group's amygdalas, but no significant changes in the hippocampal regions of the brain for this control set, leading us to draw the conclusion that most of the differing hippocampal development happens at the late gestation period and within the first 2 weeks of life. Changes in the amygdala gene expression continue to increase until stabilizing about 4 weeks after birth. We thought that most of the phenotypic differences among the control groups was due to the hippocampal changes in the rats, but now we know that these changes are also linked to the amygdalar development as well, being that the amygdala is the center for fear and stress responses in the brain. Using scientific literature, we know that the lesions in the amygdala of P7 rats cause juvenile lack of play, lack of sociality in adult rats. But in our experiment, we found that there was an increased need for amphetamines to enable movement, a decreased amount of dopamine receptors in the nucleus accumbens, and even a greater number of spontaneous dopamine releases in the brain. We know that in the hippocampal regions of the brain, there's a rapid expansion of the dendritic system for the first 3 weeks of life. In altering these parts of the brain using the P7 and P14 rats, there's a large disruption in the neurogenesis in the axons of the nearby neurons that are not able to properly expand and connect to other dendrites. Disruption in these neurogenesis within the first 2 weeks of birth can lead to lifelong neurological consequences, including lack of novelty, regulation of neurostress circuits, and controlling anxiety and fear-like behaviors. When looking at the gene expression changes between the two control groups, we chose to focus on the COX enzyme, being the enzyme that is the limiting factor for ATP production. In those brains with higher COX activity, there's a proven increase in synaptic terminals and firing within the hippocampus. There was a noticeable difference in the COX activity among the LR and HR rats in the amygdala and in several regions of the hippocampus in the LR rat brains. We noticed the primary differences in the neurodevelopment and the COX activity were among the rats in the P7 data set, in which the differences were noted in some regions of the hippocampus and the amygdala in the LR rats. Even though there was a greater change in the gene expression for the amygdala in the P21 rats, we concluded that the regions of the brain that were affected can strengthen over time, whereas the rats affected in the P7 data have less correlation because the two regions of the brain after being altered so early after birth. When examining the LR and HR rats, we determined that the COX activity and changes in the hippocampal and amygdala regions of the brain have a great effect on psychiatric disorders in the rats. But environmental factors also play a role in the rats. They were specifically bred to be HR or LR rats and also differed in maternal parenting style and anxiety depressive-like behavior before the experiment was even conducted. A future experiment could be to alter the genes within the amygdala using LR-HR rats and even perform the same experiment without selectively breeding the rats to be LR-HR rats prior.